Okay, so welcome everyone. We'll have two speakers in this session talking about proteomics. And the first speaker will be Christophe Vanderaa, who is a PhD student at the Université Catholique de Louvain. And he will talk about standardized and reproducible analysis of mass spectrometry-based single-cell proteomics data. He'll be sharing a pre-recorded talk, and I will play the video for you.
Hello everyone, thank you for attending my presentation and thanks to the organization for giving me the opportunity to present my work today. So I want to talk about a standardized computational framework for the analysis of mass spectrometry-based single-cell proteomics data. So as you may already know, single-cell omics are a set of techniques that generate measurements for one cell. This allows you to model in more details confounding effects, such as the presence of different cell types, as illustrated here on the left, or the presence of continuous effects, as often observed in developmental processes. The single-cell RNA sequencing is probably the most popular single-cell technology, and it has matured very quickly over the last few years. Several standardized analysis pipelines came out, and I highly recommend the Bioconductor book about orchestrating single-cell analyses. It really helped me digging into the field. However, when you are interested in cellular function, you would rather look at proteins rather than RNA. You might think that RNA is a good proxy for protein, and I would say, well, yes and no. If you look at the global scale, so across tissues and across genes, you can say there is a good correlation between RNA and protein. See here, each dot represents the expression of a gene in different tissues at either RNA level or protein level. However, if you focus on a particular gene, so for instance, gene 2 in red, well, you can see that the protein level is not well explained by RNA. So looking at protein and RNA gives complementary but different information. Popular tools for acquiring single-cell proteomics are flow cytometry or CytSeq. They are excellent tools and they provide high throughput and high sensitivity. However, they have the drawback that they rely on antibodies. So the acquisition can only detect the targets for the antibodies that you have. And antibodies can be subjected to specificity issues. Therefore, we might be interested in getting shotgun proteomics data, a bit like for single-cell RNA-seq. But is it actually possible to get those protein measurements, since you think about single-cell RNA-seq is relying on PCR? Well, according to this graph, it is possible. See here, we have this is a distribution of the copy numbers in a single cell for either RNA in blue, protein in red, or peptides in yellow. So peptides are fragments of proteins. So it is known that RNA is present at very low copy numbers, and therefore it is essential to have PCR to be able to measure those copies, to amplify the copies and be able to measure them. On the other hand, proteins, they are between 1 and 2 orders of magnitude more abundant than RNA. This allows to decrease the counting error first, but it also means that we could potentially measure them using mass spectrometry. And this challenge was tackled by different groups, and several amazing developments have emerged in the last three years. Recently, the Slavov lab has reached a milestone with their Scope 2 protocol by sequencing about 1,500 single cells. And this they could quantify 3,000 proteins with an average of 600 proteins per cell. Here is a general overview of how mass spectrometry works. So first, the proteins from isolated cells are first extracted and digested into peptides. The peptides are then separated by liquid chromatography based on their chemical properties. The separated peptides are then analyzed by a mass spectrometer that generates mass fingerprints. This is called spectra. The spectra are then matched to a reference database to retrieve the peptide sequence. And when a spectrum was effectively matched, we then call them peptide to spectrum match. And they can be quantified based on the signal intensity. So I should tell you here that due to the current technical limitation, at most 10 single cells can be acquired per run. We'll see the implication of this later. So, mass spectrometry-based single-cell proteomics is an emerging field that requires new computational developments. And we want to tackle here the lack of good computational tools for handling such data. Our contribution lies in 3 parts. So first, we have developed a data framework that combines existing bioconductor classes. We also provide the SCP data package that disseminates curated SCP datasets for method development and benchmarking. And unfortunately, I didn't manage to get this done for the conference, but this should be soon available on Bioconductor. We also provide another package called SCP that implements functions to streamline the analysis of SCP data. And this is already available on Bioconductor. Okay, let's first talk about the data framework. So we are interested in single-cell proteomics, so we need tools for single-cell and for proteomics. So from the single-cell part, it seems obvious to use the single-cell experiment package. It provides a compact data representation where quantitative data can be retrieved along with genes and sample annotation, as well as single-cell specific features. On the proteomics side, well, Laurent has recently released the Q-features class and package. That is an extension of the multi-assay experiment class. So remember, the acquisition starts from proteins that are digested into peptides and then measured as PSMs, while the idea is to go the other way around and to computationally, to reconstruct the protein information from the PSM data. So the different levels of informations are stored in separate objects, and the relationships between the rows are preserved. So here, for instance, if we look, different PSMs can be mapped to a single peptide, and then several peptides can be contained in this, can be part of the same protein. And when we combine, combining those two classes, those single-cell experiment and the Q-features, we get our SCP framework. The SCP framework is therefore a Q-features object that contains single-cell experiment objects. So the PSM, you can see that at the PSM level, there are several assays, because single cells cannot be acquired all in the same batch, and so we keep them in separated assays, and those assays can then be integrated together and aggregated to peptide data. The peptide data can then be aggregated to the protein data. And see again here that the relationships between features are preserved. So this is very nice conceptually, but how can you see this in action? Well, let's get some examples. So you can find examples of published single-cell proteomics datasets in the SCP data package. It currently contains 11 datasets that are accessible through Experiment Hub. So I want to give a little shout out to Experiment Hub, to the Experiment Hub project, as it makes disseminating data super easy. So just look at this. all you need to do, so if let's suppose you want the scope 2 data set, all you need to do is load the SCP data package and then call the function spect 2019 v3, and you get the data, and Experiment Hub will take care of caching the data for you. When we look at the overview of this scope 2 dataset, we can see that it indeed is a QFeatures object, and it contains multiple assays. So the 177 first assays are PSM data in the different batches, and the last and the before last assays are the peptide and the protein data. So see here that each assay is indeed also a single cell experiment assay. So now the idea is from the PSM data trying to reconstruct this peptide and this protein data set. Yeah, this was already done here by the scope 2 and provided by the authors, but it might be interesting to be to test our tools and do this by ourselves. And so here is what is shown. On the left, you can see the scope 2 pipeline, the different steps that they took to build this peptide and protein data. And on the right, so yeah, I should say that this code is actually, well, the code they used to do this, they built almost everything from scratch, and it was difficult to use, and hence it's not possible to apply to other data sets. So we took the effort to standardize all of this, and the result is on the right. So I should say that most functions are actually implemented already in the QFeatures and the multi-assay experiment packages. But SCP is actually an extension to provide single-cell proteomics specific functionalities, such as, for instance, reading in the data or computing specific single-cell metrics. So I hope you will agree with me when I say this code is easy to read and that you don't need to be an expert in R to get a general sense of what the code does. And furthermore, tweaking steps and methods in this script becomes very easy, and you just have to change either the order of operation. You can remove a step, or you can just change an argument in one of the methods arguments to simply adapt the pipeline. So we applied our tools to reproduce the scope 2 analysis, and look at here what are and we can compare those two results. So first on the left, so there are, if you notice, there were different filtering steps. So do we get the same results out of those filters? So if we look both at the cells or the proteins, we can see there's almost a perfect overlap between the two data sets. So meaning that we could correctly reproduce the filtering steps. But okay, it's nice to have the same filters, but do we have actually the same data? So if we take all the common proteins and the common cells between the two data sets, and that we subtract one from the other, we can get a sense of, okay, what are the quantitative differences? And you can see from the plot in the middle that there is a very sharp peak around 0, so meaning that most of the data is actually the same. But there are some slight differences, and maybe those differences can distort the patterns that were observed in the data. And namely, one pattern that is of interest in the article was to separate monocytes from macrophages. And this is shown on the top right. So you can see they had some custom PCA, and they could nicely, well, not perfectly, but nicely distinguish, separate macrophages and monocytes. If you look at the bottom right plot, this is what we get with our SCP tools. And you can see that basically the pattern is conserved, and we could clearly reproduce the data. So currently we use our tools to reproduce two data sets. I just showed you one, the scope 2 data set. So reproducing is nice, and especially when you can identify issues and suggest improvements to the analysis. Then we tried reproducing another data. This time, so it's a bit it's a technical detail, but this data set was acquired with label-free quantification protocol. And in this case, we actually could not replicate the results. But I want to say this is not a limitation in our software. This is rather because a lack of good documentation did not allow me or allow us to do the same thing as they did. Yeah. So I think this can demonstrate the successful application of our software to different types of single-cell proteomics data sets. And again, I wanted to have this done for this conference, but this not happened either. But I'm currently writing a preprint, and this should come out soon. Okay, so here I am now. I'm geared up with my different single-cell proteomics tools, and there are some ready to tackle some interesting challenges. So the challenges are actually, well, common to any single-cell technology. So first, there are technical challenges. The methods should be automated. It should be able to handle minute sample amounts, and the cost per cell should be very low. So this is not really my part. I'm more interested in the computational challenges. So single cell data, as we have many samples, becomes you can quickly get big data. It's also characterized by high missingness and by complex batch effects. And I think there's also another interesting challenge in single cell technologies is the conceptual challenges. For instance, what is a cell type actually? And where do we put the border between cell type and cell state? Or for instance, what is biologically interesting? We usually regress out single cell cycle effects, but are those now becoming biologically irrelevant and might depend on some biological processes? But yeah, this is food for thought. Coming back to the computational challenges, so I talked about missingness and complex batch effects. This is true for other techniques, but I want to show you more in practice what those looks like on single cell proteomics data, and again taking the scope 2 data sets. So first, batch effects. They can be caused by different factors. So first, sample collection. We collect for technical reason, different batches can be samples can be collected in different batches, but there are also batch effects linked to the instrument. So for instance, we can have chromatographic batches because when running different samples at the moment, we need to change the chromatographic column, or the mass spectrometry. At the moment, mass spectrometry must go on the maintenance. Detectors must be calibrated, and this leads to differences, as you can see on this graph, where you have, in this case, 4 different chromatographic batches. Well, the color indicates a chromatographic batch. This type of batches, for the moment, were removed using the combat algorithm, but probably and maybe other better solutions could be found. Then we have missingness. So missingness, you can see here, most of the, sorry, I should explain this graph. So each dot here is information for one peptide. On the x-axis, you have the missingness of that peptide in macrophages and the missingness of that peptide in monocytes. And you can see that generally for both cell types, the missingness is around 95%, something like that. However, something interesting, and those 95% are, of course, due to technical missingness. But something interesting is there seems to be also a biological component to this missingness. And if you look, for instance, at this point, you can see that it's more missing in monocytes than it is in macrophage. And the color gives you the log4 change, and red means more expressed in macrophage. So it's interesting to see when it's more, because it's more, it is more expressed in macrophages, and maybe therefore also less missing in those cell types. So finally, okay, actually batch effect and missingness are two things that are combined, and this is what we observe in single cell data. So I show you here 3 different runs. On rows are different peptides and columns are different samples. And you see several samples have been acquired in the same run. And we can see just blocks where some peptides were not identified in one batch, but they were in another. So I want to conclude my presentation with some take-home messages. So first, single-cell proteomics is an emerging but very promising field. We have developed a computational infrastructure to formalize single-cell proteomics data analyses. I showed you that we could apply and successfully reproduce one published analysis, and we also reproduced a second one, but could not replicate the results. And I think I showed you that there are still some exciting challenges that are to be solved. And I want to just conclude saying that I see the work, so the single-cell proteomics framework currently as a bridge between two big fields, so the single-cell field and the proteomics field. And for the moment, you can see that this bridge is, I think, clean and neat, but still very simple. And I hope that with collaborations, with more developments, we can make some fancy bridges with nice towers and maybe have something looking like the London Bridge. Yeah, that's my dream. Okay, so just I've finished with some acknowledgements. I want to thank my lab, the CBio lab at the UC Louvain, and I want to namely thank my promoter Laurent Gatteau. I want also thank the Slavov lab, Nikolai Slavov and Harrison Specht, for their input for reproducing the Scope 2 analysis. I also want to thank the bioconductor team, and namely Laurie Shepherd, for valuable help on putting SCP data on Experiment Hub. I want also to thank the bioconductor team community and the Eurobio community for those excellent developments and packages in R, and for this amazing conference. And of course, I want to thank you for your attention, and I'm happy to take any questions. Thank you.
Thank you, Christophe, for this very interesting talk on this very complex data, for sharing your dreams about bridge building. We already have one question from the crowd, from Nils Kurzava, who's saying really great work, which is absolutely crucial for the development of the technology. And did you have a look at what effect different imputation methods or no imputation have on the results of the analysis?
Yeah, so I think that's really a great question, and I mean, I have no clear answer on what is the best thing to do regarding imputation. Maybe what I want to say is, so for the Scope 2 analysis, imputation was performed using k-nearest neighbors. I don't know if that's the best solution. In let's say on the bulk proteomics, there have been some indication this is good, but is this good for single cell? I don't know. And I would actually say I'm not sure imputation is a good idea, because if you think there's over 90% missingness, and imputation is basically predicting values that you don't see. So do we really want to focus all on analysis on predicted values, knowing that there potentially might be some artifacts? I don't know. And clearly we can see that before imputation, if we look at the batch effects, so in this case, batch effects were corrected with combat, which is not robust against missingness. But so we see in the PCA in a missingness safe PCA, so with nipples, we can see that there are batch effects in the first principal components, but in the principal component 4 and 5, we can kind of reproduce the shape I showed you, where two cell types are separated. So it seems that even before imputation, this data, the underlying patterns are there. So I'm not sure PCA is useful. Maybe a correct batch effect might be better, and modeling the missingness in the thing.
Yep. Okay. Thanks. Maybe we'll take one more question before we move on to the next speaker, and then we'll. So any remaining questions, I'm sure Christophe will be happy to answer them on the platform.
We can discuss this on the tables.
There's one more question from Etienne Becht, who's saying, really interesting talk. Do people ever run unsupervised analyses directly on the spectrum without first mapping them to specific proteins?
I must say this. I think I've read this somewhere. I mean, in single cell, definitely not. This is, I'm sure. Maybe in bulk proteomics, I think I've read this somewhere. But yeah, I have no experience with that, and it's like a vague memory, so I cannot give informed information on this.
Okay, thanks.
Yeah, you're welcome.
Cool. So the next speaker will be Oliver Crook, who's a postdoc at the University of Oxford, and he'll be giving a live talk on the effect of phosphorylation on the spatial proteome.
I'm Ollie. I want to talk to you about the effect of phosphorylation on the spatial proteome. This is work that I partially did in my PhD, the Cambridge Centre for Proteomics, and I want to start off saying this is joint work with Anya, who did a lot of the mass spectrometry sample preparation, development and stuff like that. So first of all, why study phosphorylation? Well, phosphorylation is a molecular toggle. It allows us to change the function of a protein without, completely synthesizing a new protein by adding a phosphate modification to some of the residues on that protein. So, for example, this might change interactions, move that protein to another location to carry out the same or a different function. Really, there are kind of many possibilities. And this process is regulated by kinases and phosphatases. And you can just think of kinases switching on that phosphorylation and phosphatase switching off that phosphorylation. The kind of canonical example of why phosphorylation is important to study is the cytokine is detected by a receptor, and this leads to downstream phosphorylation of STATs that then translocate from the cytoplasm into the nucleus to then regulate a number of transcriptional programs. Phosphorylation isn't the only post-translational modification. There are many, just a few indicated here, that essentially allow millions of proteoforms within the cell, each carrying out a bespoke function. And what do all these post-translational modifications do? Well, even kind of the best studied ones, such as phosphorylation, we're only really starting to unpack what are the actual functions of these phosphosites or phosphorylated proteins. Recently, a group used machine learning algorithms to develop a score. So given a phosphosite, how likely is it to be functional? It's not even clear that any particular phosphorylation is even interesting. Several groups simultaneously have shown that certain phosphorylations can affect the thermal stability, so increase the melting temperature or decrease the melting temperature of a protein. And several groups or other groups have shown that phosphorylation can also mediate protein-protein interactions. So interaction can only take place in a phosphorylated state. And particularly, they pointed out this is particularly common for RNA binding complexes. But I want to talk to you about phosphoproteomics and its spatial relationship. So first of all, how do we perform spatial subcellular proteomics? There are many methods, and just to give you an idea, so one of the canonical example is microscopy, where you just look under the microscope and see where is my protein, but this requires an antibody, so you are at the will of the specificity of that antibody. It's low in throughput and can be rather expensive. We have a further challenge in phosphoproteomics because you need an antibody that's specific to the phosphorylated version of that protein, not just that protein. Another set of tools for spatial proteomics is proximity labeling, in which you use a bait protein to catalyze the biotinylation of proteins in proximity. And then you can use several baits across the cell to kind of give you a complete picture. But again, this can be very tricky to do, requires a lot of protein engineering, and in general suffers from false positives, and it suffers from a lot of the similar problems of being low in throughput. So how do we perform high throughput spatial proteomics? Well, we start off with a population of cells, and we're going to lyse them gently to keep the internal organelles intact. So then this lysate is going to be subject to increasing centrifugation speeds, and each fraction will be differentially abundant for different organelles based on their biochemical properties. What does spatial proteomics data look like? So this is just a PCA plot, the first 2 dimensions. Each pointer is a protein. Here we're measuring roughly 5,000 proteins. Some proteins are colored. These are markers, so they're well annotated proteins where we know their subcellular localization before we started this experiment. And as you can see, we resolve a large number of subcellular components, that's small and large ribosomal subunits, nucleus, chromatin-associated proteins, mitochondrial proteins of different elements of the secretory pathway. And those proteins where we don't know the localization, we want to assign it to one of the particular localizations. So these are the profiles from those markers that we saw before. And we can do this using machine learning algorithms that are implemented in our bioconductor packages. So it's basically, simplistically, this is association by correlation, and in reality, something more complex is happening. Now, how do we extend this framework to capture spatial information on phosphoproteins? So we're going to take 10 fractions submitted to increasing centrifugation speeds, again, differentially pelleting those organelles. They are going to digest 2 proteins. We're going to label each of these samples with TMT, and you can just think of these as barcodes for each of these samples. These are isobaric barcodes, so just look exactly the same inside the mass spectrometer. We'll then multiplex these into a single sample. Now, to obtain phosphorylation information, you can use a kit, but we're working with a particularly complex set of data. So we want really deep coverage. So there's a kind of triple enrichment strategy that we use to obtain really high quality phosphorylation steps. So it uses titanium dioxide treatments, but also chromatographic enrichment strategies. And these phosphoprotein samples, along with our total protein samples, are subject to high quality, high accuracy quantitative proteomics via mass spectrometry. So what do our data look like? So all the circles, as before, are our proteins. Again, some of them are colored. These are proteins with known localizations. And I've drawn little snowflakes, and these are our phosphoproteins that we measured. As you can probably see, there seems to be an enrichment in the kind of top right-hand corner of the plot, which is not unsurprising. These are the kind of nuclear and cytosolic regions where we expect phosphorylation to be quite high. And we see only a few points in this kind of mitochondrial region where succinylation is a much more important post-translational modification, and phosphorylation is rarely seen. Again, this is all done using our bioconductor packages. So we can then allocate those phosphopeptides to different subcellular localizations. So for example, a few hundred proteins to the nucleus, very small number of proteins to the mitochondria, maybe half a dozen or so to the ER. Now, some phosphoproteins have a different localization to their parent protein. So for example, the parent protein might be located to the cytosol, and when it's phosphorylated, it translocates to the nucleus, or simply a nuclear pool of that protein just happens to be phosphorylated as well. And we want to detect these samples or these particular proteins in our sample because they're interesting. We think they're more functional. And so how does this appear in our data? Well, these proteins would have different profiles or different trajectories across those fractions, so different abundance and distributions. So we want to test for this across a whole list of phosphoproteins. How are we going to do this? Well, we're going to develop a Bayesian two-sample test, and we're going to weigh up whether a simple shared trajectory, so the same pattern for both the modified and unmodified version of the peptide are the same, versus an independent model where the modified and unmodified version of the peptide have different profiles. And this is indicative of a change in localization. Now, in the Bayesian framework, we're going to take this as a model selection problem and call this kind of model 0, model 1, and we want to weigh the relative plausibility of these two models given the data. And you can think of this as you compute this as the posterior probability of this independent model given the data, or maybe you prefer to think of this as some kind of posterior odds of these two models. I haven't yet told you how we model the data. So we're going to, our response is going to be the abundance or normalized expression, a number of linear predictors based on centrifugation speed, a smoothing term, plus some noise. Now, quantitative data at the peptide level is very noisy, much noisier than at the protein level. So we're going to model our residuals in a robust way, allowing t-distributed errors, and we're going to encourage shrinkage, priors that encourage shrinkage as well. We're going to shrink our coefficients, similar to how you would ridge regression-based analysis. And then we're going to use a number of a non-parametric term to scoop up any of the residual correlation that's not well modeled by a linear model. So how do these data look? So just to see, this is the protein, this is the triptych peptide that we're testing and have measured, and this is the modified residue that we're interested in. And as you can see, for this peptide, the modified and unmodified trajectories are different, and this is indicative of a change in localization for this peptide. Now, we identify just less than 100 proteins where they have a differential localization upon phosphorylation, and we compare these to the functional score of this Nature Biotechnology paper. And in general, we found that our differentially localized phosphoproteins had a 10% higher functional score. Now, I could simply do a test to see if these distributions are different, but that's not that interesting. What we want to know is if I do another experiment and maybe a shotgun proteomics experiment, and I measure a particular phosphoprotein and it's active, should I follow up and do maybe some microscopy work? So we wanted to know if these scores were predictive of differential localization. So we fit a logistic regression model, and obviously increasing scores lead to increasing probability of differential localization. The signal is weak. Obviously, not every functional phosphopeptide is a change in localization. There are many others, but this could be useful for other people as well. Some examples we see in the data, so one we're particularly interested in is ERK. We're very fortunate to measure this triptych peptide and a phosphorylation at site 187. This is curious because this falls in exactly the kinase activation motif for this protein. Unfortunately, we don't have much localization information here or in the Human Protein Atlas. It kind of floats in a little bit in the middle of nowhere. But we did manage to obtain that the phosphoprotein is differentially localized. So we try to follow up on this, and this kind of correlates with the fact that known interactors, RET is localized to the cytosol, and JAK2, another interactor of this protein, is kind of very confidently localized to the nucleus and also the plasma membrane occasionally. Another example, a little bit more obscure, so Celanon, It's a protein that regulates calcium homeostasis of the ER, so it's an ER localized protein, but it can also be found in the cytosol, according to the HPA. And we measure phosphorylation in exactly the ER signal peptide for this protein. You can clearly see the modified and unmodified versions of this peptide have different trajectories, suggesting that this phosphorylation in this signal peptide is really key for the localization of this protein. So just to summarize, phosphorylation is a key molecular toggle, but it remains largely uncharacterized. High throughput spatial proteomics can obtain localized information for thousands of phosphopeptides. Some phosphopeptides have differential localization compared with their parent protein, and higher functional scores are suggestive of differential localization. And thank you for listening.
Great, thanks, Oliver. That was a really great talk. So we're still happy to take questions. So if you have any, please type them in the Q&A. So I'll start with one. So you talked about differential localization, and we saw these nice plots on whether being phosphorylated or not impacts the localization. And you mentioned you're looking at some kind of normalized abundance, and I was wondering how these data are being normalized.
Yeah, so first of all, we have to correct for the input of each sample. So there's some kind of median normalization that needs to happen. And we correct the phosphorylation data based on sample inputs at the total protein level to avoid any bias. Yeah, there's a kind of log transformation because these data are kind of log normally distributed mass spec stuff. Yeah.
Great, thanks. So there's a question from Nils Kurzawa, who's saying it was a great talk, and did you happen to find any overlapping sites with the phospho-TPP?
Yeah, that's a good question. So we have looked at that. Interestingly, the overlap of phospho sites that we measured and the phospho-TPP are actually quite small. And so there are a few overlaps, but we didn't kind of see any statistical enrichment. And we wonder whether this is the phosphorylation that affects a protein stability is probably orthogonal to the phosphorylation that affects localization. So we didn't see any statistical overlap, but we agree that it's functional. It's interesting that it seems to be quite orthogonal. We did see quite a lot of overlap for the complex dependent phosphorylation. Our worry here is that we have a kind of strong enrichment for kind of nucleus and cytosolic proteins where these RNA binding proteins are in general localized. So we're kind of unsure if that interpretation is a kind of real functional enrichment or just a kind of side effect of where we're actually measuring these.
Great, thanks. There's no more questions for the moment. Maybe I'll ask a quick one. So you mentioned that phosphorylation is not always functional. And can you give some intuition? Do you have an idea on why some might be functional and others aren't? And why would the phosphorylation then be useful?
Yeah, that's a good question. So we've identified, many people across many different experiments have identified kind of hundreds of thousands of phosphosites. And the question is, how is these processes being regulated? Is the phosphorylation being, put on by a kinase and then taken off by a phosphatase so quickly that it's not really there? It's just kind of random chance. When you might only find that some of these phosphorylations are active in particular cell states, so you might have to stress the cell in a particular way before you find a particular phosphorylation to be functional. We're not looking at any particular perturbation, no heat stress or osmotic stress or anything like that. So it may be that some of the phosphorylations that we're not functional or not found to be functional are functional in particular scenarios that just haven't been captured yet. So kind of many reasons it's hard to unravel them all.
Yeah, I think that makes sense. There's still a lot to learn. All right, so thanks to both the speakers of this session. I wish I could give you a real applause. So we'll end the session now. Thanks everyone for joining.